זה בעיה. כי ברגע שהוא הדליק נהיה לו אנטארקטיקה. אבל כאילו אין מה לעשות, כי זה עניין שהוא בהגדרה בינארי. כן? זה לא on-off. זהו. סבבה, אבל אולי הוא לא צריך שיש לנו אוקיי. רעיון.
Good morning, everyone. Let me take this. Okay. So, good morning. This is my last session with you, and what we're going to have is me going for about a half an hour just to try to complete what I have started in the last lesson, which is something about object recognition and the ventral stream, and give you some idea about sort of processing that's done along the ventral stream. And then we'll take a break after half an hour. So time me after an hour, whatever, we stop, we take a break for 10 minutes, and after the break we have the students giving their talks, each for something like 30 minutes or so. Okay, so that's the plan for today. And I want to just get back to the sort of slide where I sort of finished last time, which was basically just describing something about the ventral stream of the information going from the retina via the LGN to V1, and then sort of moving along the ventral pathway. And that was just a sort of a summary of the how many neurons do you have in each area? What is the output of these neurons? So let's say there's 190 million neurons in human V1, and 37 million of them give input to V2, and so on and so forth. This is the latency of response. So as you move along the hierarchy, you can see that the latency of response increases significantly from something like 40 milliseconds to 100 milliseconds in anterior and infratemporal cortex. And here you see this is the dorsal stream that we talked about. We talked about MT and LIP in the previous lecture. So today we're going to focus on the ventral stream. And then that's another way of illustrating the same thing. That's just something about sort of the complexity of the stimuli which drive the cells along the ventral stream. So again, going from V1, and just to make things complicated, you see here TEO and TE. So TEO corresponds to posterior IT, and TE corresponds to anterior IT. The reason is that different anatomists use different notations just to make God knows why. Anyway, and then we talked about, so of course, we extensively talked about neurons in V1 and orientation tuning, and the fact that you have sort of a Fourier analysis, possibly Fourier analysis via these Gabor filters that we mentioned. And then I briefly mentioned that as you move along, you see, for instance, in V2 cells which are responsive to subjective contours. So they'd have a receptor field here, and they'd be orientation tuned to this orientation, to the oblique orientation here. While there's nothing inside the receptive field, of course, we see this as sort of as a rectangle oriented at 45 degrees. So the cell would be sensitive to that. And then as you move along, you may find cells in temporal cortex, in posterior temporal cortex, and anterior infratemporal cortex, which are sensitive to more complex stimuli. And this is just a depiction of how activity spreads along initially from V1 and MT, interestingly. Further on, as time passes by, you get more and more activation of cortex. Notice also that there's activity initially in the dorsal stream, MT, and then in frontal eye fields, which are related to eye movements, and only later in the ventral stream. Okay, so that's where we ended up last time. And I'll just fly through this to give you some idea of, okay, what do we think happens beyond V1? So the first thing to notice, that's a token, I won't have time to get into a lot of details, is that you find these cells, for instance, in V2, which signify borders. Okay, so what do you need to do? Of course, you have an image such as this one, and the first thing that you have, typically, is to sort of segregate figure from ground, decide what's part of the figure, and segregate the different objects, right? And that's a very difficult task, because if you think about it, the neurons in the retina and LGN or in V1 are just sensitive to contours, okay? But what part of the contour belongs, or which side of the contour belongs to the figure and which side belongs to the ground? That's a more tricky issue. But you do find cells like this already in V2, okay, the next stage. So here's just an example this is from Fonterheit's work. So you see here's the receptive field of the cell, and you see if the figure, that is the square, is on the left side, then you get nice response. And it's contrast insensitive, right? So it doesn't matter if it's white and with a rather grayish background, or rather black on a whiter background. In both cases, you get strong firing, while if the figure is on the right side, you don't get much of a response. Okay, so here's something about border ownership, which you can find in V2. And of course, the idea is that if you pick these borders, you can start depicting where the figure starts and where it ends, without going into much detail about this. And generally, there have been a few sort of suggestions for models, which I won't have time to get into, but that's called an H-max model, which suggests that you have some, first of all, convergence and sort of maximum rule applied to this, in which you sort of go from neurons which are oriented, just oriented to the curves, that you sum up information, and what you see here is just sort of trying to follow what we know about the pathway going from V1 to anterior IT here. So the receptive fields, as you can see, become bigger. Interesting thing is that all this is sort of, this model is such, as I said, without going into detail, is that you sort of sum up the information from the earlier area, and this sort of determines the receptive field of the next stage, okay? And the interesting thing is that if you can see here, all these stages up to the classification. So the job of this model is to say, okay, is this an animal or non-animal? We're going to hear about that a little later in the talks. Okay, so here's these different images, and you have to classify, is it animal or non-animal? So, okay, at the last stage, where we have these classification units, which notice they're very small, okay? So you can see here the numbers of units in each level, okay? So the classification neurons are a very small number of units. But here what you have is supervised task-dependent learning, meaning that you change the sort of the strength of the inputs from this level here to here via a learning rule, which is basically supervised. Supervised means that okay, each time you made a correct answer, you get you are sort of notified, while in the unsupervised case, there's no sort of error signal telling you if you did right or did wrong, okay? Now, the amazing thing about it is, think about this. So, first of all, a few points here. First, this is a completely feed-forward network, okay? Complete feed-forward network. Two, there's supervision, which is different from us, right? We are not supervised as infants for each image, is this a tiger or is this an animal or non-animal, right? I mean, so we learn this in unsupervised way, so this is non-biological in that respect, but all the other levels are unsupervised. Okay? So there's a lot of details here. I don't have much time to get into this, but just to give you an idea that, okay, there have been some models that have been developed, and that's something maybe 10 years ago to sort of get some classification. And of course, what I'm not getting into, but you probably will hear, is the deep neural network models, which have sort of exploded in the last, I don't know, 8 to 6 years, okay? And they're basically the same, just the thing is what has changed is that there's much more levels, right? So there could be 100 layers, 30 layers, 100 layers, okay? And they're getting better and better. You know, this is what's been used now, for instance, by Amnon Shashua to decide to have a car navigate on a busy road and avoid having accidents, right? So again, the idea is that you have these layers, and then you may have 30 of such layers, or so, tens of layers, and what you get is sort of on each layer, you have sort of a learning rule which originates from the highest level and sort of propagates back, okay, so that you change the weights slowly. And the point there is that you use statistical learning. This is different from here, but you use their statistical learning so that you basically learn from exemplars. And the exemplars could be a few hundreds of thousands of such exemplars, typically videos, in which you label the image and say, here's the car, here's the bicycle, here's the signpost, and so on and so forth. And once you do that, okay, using statistical learning techniques, you can generalize, okay? But the point is, it's very different from human learning in a sense that it's supervised, okay? So there's a human basically labeling the images, and then if you have enough data, and you really need a lot of data for that, you can generalize, okay? And they're very efficient. As you know, statistical learning algorithms have been used now not just in object recognition, face recognition, whatever, what you want, okay? And they're very successful, but they're very different, okay, from the way we learn, and they're limited in the sense that you need a human sort of teacher sort of to learn from exemplars, okay? So if you provide the answers from the exemplars, they generalize very well. All right, that was just a shortcut. I go back to physiology. I want to describe a little about what's happening in higher order areas. So we talked about V1. I just mentioned to you about V2, about these subjective contours, border ownership. And now moving on to sort of areas. So this is anterior IT here, just to give you the flavor of this. So when physiologists started to record from this, and this is recording from awake monkey, they were quite puzzled, okay? So the question is, I want to raise to you is, how do you start dealing with this, okay? So when we were in V1, what you could use is sort of a basis functions of these orientation-tuned neurons. And Fourier is a very good basis function, right? So you have the different frequencies and different orientations. And one can prove that, with sort of filters of the different orientations and different frequencies, you can represent, that's what Fourier showed, you can represent any image, correct? So it's a sort of a complete basis function describing, the energy of these different filters. You can now rebuild what the image is. So now the question was, okay, when you get to sort of IT, what stimuli should you present to start with, okay? And that's just to show you the puzzle of it. Okay, so here's just, okay, so this was, I think, work by Nikos Logotheris and David Scheinberg, in which they just use images of, I don't know, a cup from the Queen Victoria's set, whatever, and sort of a cat, a goose, whatever, what you want. What you see for yourself is, okay, what is the cell sensitive to? It's a rhetorical question. I don't know. It's hard to tell. Okay, it responds as well to this cup here, but it also responds as well to the bear here or to the cow, right? Okay, so it doesn't seem, I mean, there's a classical case where you can't say it's a bear neuron or a cow neuron, or, right? It seems, okay, so what are the alternatives? What could possibly be the case? Well, one case that I can think of, it was a question to you guys, but since it's early in the morning, I'm answering. One thing you can think of is, well, maybe there's a contour, okay, say, I don't know, here, the back of the cow, which is also found in the cup here, but not here and not here. Oh, sorry, also here, okay? So you can look at, I mean, that's one option, just look at the overlap and see if there's just this, is there some underlying simplified principle that generates the activity. But when people looked at this, that was not the case, okay? So what's the alternative? The alternative is very much related to what you're going to hear today. So how should we start thinking about this? So now this is to the speakers of today, because it points to the same thing. I mean, so you can think about this. Think about a vector representation. You're not representing an object, you know, with, I don't know, grandmother cells. So every time you see grandmother, there's 5 grandmother cells which fire, or 1 grandmother cell which fires, or a bear cell when you see the white bear or something like that. No, don't think about this in this way, but think about this as sort of a representation across n neurons. We don't know how big n is, but a large set of neurons. And then you can start thinking about a pattern of representation. I'll get back to that. I'm just sort of, I want to move on, but I think that you shouldn't think about this as sort of a feature detector. It's not a bear. When you get to IT, I don't think you should think about neurons as a bear neuron or a cow neuron or a whatever. Okay, so that's the first thing to keep in mind. Now I want to move a little backward to posterior IT, where the input gets to anterior IT. So one person who did this in the 90s, early 90s, was Keiji Tanaka. And it was, again, thinking about, okay, let's try to simplify stimuli to see at what point, what are the cells sensitive to? Okay, so what he did was the following. It was the early days of sort of digital imaging. So he, being a Japanese, he had the first, I don't know, a digital camera. So he took pictures. Okay, so what he did was the following, okay, literally. He had his, I think, kids were of young age, so he went and grabbed toys from the kids and just showed them to the monkey, one after the other, okay? So it just shows literally the objects themselves and records the activity. Okay, then he found, let's say, I don't know, the 20 or the 10 best objects that elicited the highest activity, okay? So here's one of them. That's sort of, I guess, a toy. What is it? The tiger, after being sort of losing some of its fur due to the kids' play. Anyway, so what he did next, having a digital camera, is first of all, take a picture, okay? So he took pictures of the 20 best stimuli, and now instead of presenting the object, he's presenting a picture of the object, okay? And then he's looking for the best stimulus among these, and say it's this tiger from an upward position. And what he starts doing now, it's somewhat arbitrary, but what he's doing is simplifying the object to see when does he get still a robust response, and when does he lose the response, in an attempt to reduce the stimulus to see what are the minimal features which generate activity in these cells. Okay, so here's just one of them. So the steps here are the steps that he's taken. Okay, so he got a robust firing here, went on to this one, so on and so forth. And then, okay, he says, okay, this is still generating activity. You can say, okay, how similar this is to this, but this is still generating activity. But if you break this down to its features or to its components, you don't get much of a response. Here's an example, a literal example of this. So he had, I don't know, this screwdriver, and then, okay, here's the firing of the cell. So now he takes a sort of breaks this down into just sort of black and white, gray level sort of a contour. He gets even a better response, right? Now he breaks this down into different options here, sort of simplifying that. And this is even stronger than this, okay? So this is sort of the stimulus which gets the biggest response, but these get a much smaller. So the numbers here correspond to what is the firing rate during the time that the stimulus is presented, and a little after that. Sort of. and you see that this is much stronger than these. And then when he breaks this down, no response whatsoever. Okay, so this is sort of the simplified stimulus that he found for that specific cell. And here's a few other examples. So you see that it doesn't care about, okay, here's the activity for this and this and this, but it does care to have this small chupchik, a small part here, combined with another object, and this small part here shouldn't be too small, because this one, the bomb here, doesn't get much of a response, neither does this, but this does. Okay, so what did he find in general? When he did this, he found that there's sort of these visual alphabets. Okay, so somewhat surprisingly, I would say, okay, that, so one cell responded best to this simplified stimulus, another to a T junction here, third one to this one, and so on. And now you can think about these as sort of letters of the alphabet. If you combine them together, okay, you may generate various objects. Okay. Now, if you talk to people from computer vision, they would tell you that this is not what they think would be the sort of reasonable alphabet, except a T junction. So T junction, I don't know if I had this. Whoops. So here's a T junction here, you see, okay? Here's a T junction, sort of signifying a sort of a change from one object to another. And that's quite often, again, I won't have time to get into this, but T junctions is something that people have expected. But the other images seem to be somewhat arbitrary. This lollipop, okay, great, but what do you do with the lollipops to build up? So when I'm saying alphabet, you see what I mean. I mean that you can combine these specific images to generate any sort of image that you want, okay? And so that was his finding. And I must say that this finding was sort of replicated, first of all, by him, to show that there are columns in IT. So just as I talked about the columns in V1, okay, and columns in MT, so remember in columns in V1 selective to the orientation, and columns in MT that are selective to the direction of motion, well, Tanaka found columns in posterior IT, and here is sort of what do I mean by a column? That if he moved his electrode along this column, he found that the cells were selective. They may be activated by this image or that image, so they're phase insensitive, but the cells along here prefer this sort of images to these, or to these, okay? And if you move to the next by column, you'd find that they'd have this sort of structure. So something, or here the T-bars that I was talking about, okay? So that was, and this was, since I don't have much time, this was also replicated using optical imaging. So when Tanaka did this originally, you just had to move your electrode and record from one cell to another and use different stimuli to figure out what was the best stimulus for that cell. But with optical imaging, what you can do is look simultaneously at a resolution of a few tens of microns at a piece of cortex. And there you could see, okay, if nearby neurons have similar preference, okay? And that's sort of corroborated in later years Tanaka's sort of columnar organization of IT. And so this seems to be holding, and this was 20 years ago, this seems to be holding. I don't have time to get into the full details of this, but this seems to be holding. Okay, so we somehow have a sort of a representation of sort of primitive features which are beyond just edges in posterior IT, which is probably used in anterior IT. Another important finding was phase cells, okay? So phase cells were found already in the 1970s by Gross, Charlie Gross and his group. And this is just a demonstration of, so what they did here was present various images, from, I don't know, from the internet. They took various images. Some of them are faces, some of them are just non-faces, different objects. And what they do, and they present them and look at the response to each of them. And they're presented one after the other in very fast speed, okay? And that's just to give you an idea that, okay, if you do these, and now you look at the response of the neuron as a function of the images. See, so I don't know, for this image, there may be a strong response, for this no response, this one little response, this one little response, so on. So you organize them according to their rank. This, so the image that elicited the highest response would be here. The image which elicited the lowest response would be here. Okay? And you sort of rank order them. And the point to notice is this is number 1, and this is number 10, and number 20, and so on. And you can see immediately it's responsive more to faces than to any other objects, right? So, I don't know, the first 50 objects are exclusively faces. There may be human faces or monkey faces. First, okay, here's the monkey face, but a human face is next to it. Okay, so, but it is responsive to faces. Okay, so this was an addition to what I talked about. So if in Tanaka I talked about these columns which may be sensitive to these T-junctions and different orientations, sort of elicited here, or these spikes here. Okay, there are other embedded in this columns which are also phase selective. Okay, so there are phase cells and phase selective columns within posterior IT. Okay. And this phase selectivity, you drew a lot of attention. People looked at this. So that there's a few clusters. There's not just one cluster. Here's another such cluster. So I'll just go over this briefly. You see there's a few of these. I don't know, 1, 2, 3. And they're given different names: posterior, lateral, anterior, frontal, anterior, medial, whatever. And you see there's about 6 different phase clusters in the monkey brain, in the temporal cortex. And okay, a few things to notice is what they've done here is sort of map them first with functional MRI. Okay, so they put the monkey in the scanner, present objects of faces and non-faces. You see, so there's these faces and other gadgets or the hands or whatever. And you see, first of all, that what you see here in the yellow are these voxels which are most selective to the faces. Okay, so after you see this, what they've done is now insert an electrode into this region and now record from the neurons. Okay, so first they sort of map which areas are selective to faces using functional MRI, and then they verify this using single unit recordings. And without getting into much detail, you see that, okay, so this is the number of cells, and you see that the vast majority of them are tuned to faces. So all these, I don't know, images 0 to 16 are different faces. And then there's different, I don't know, fruits, and then the gadgets, hands, and you see that this is, you can see the column here showing that they're all sensitive to faces, not to other objects. They're not very sensitive to an individual face, because you see that they're all, the dots here are spread. So each line here corresponds to one neuron, if I wasn't clear about this. And you can see that there are some neurons here, like, I don't know, maybe 10% of the neurons which are not phase selective, but most of the neurons are phase selective. And another reason I'm showing this is to show that, okay, this is also proof of how fMRI is such a good and sensitive techniques. Because if you were to record something using fMRI, it had nothing to do with neuronal responses, then if you were to, okay, remember that they're moving their electrode to the sites according to the functional imaging data, okay? And the fact is that they verify this because the vast majority of the cells are phase selective, okay? Sorry about that, didn't mean this. All right, and then, since I don't have much time, I'll just say that, okay, and you see these various areas. Well, what's happening in these different areas? They're all phase selective, yes, but there's something different. You expect that something would be different between these different areas here. So this is shown sort of here. And the basic thing to notice is that, okay, what about orientation of the face? So you may see me like this, or like this, or like this, and different, and you recognize it's me, okay, irrespective of the orientation of the face orientation. So they looked into this. Just to show you, before I get into this, is that you see, okay, here's the responses to the faces, but it's enough also to have just a cartoon of the face. So you see, once you preserve the eyes and the nose and the mouth and the general shape of the faces, you get a great response of the cells. But if you sort of eliminate the eyes, or the sort of, and you're left with this, I don't know, horseshoe, you don't get much of a response. So there's sort of limited information in this about faces, of course. and therefore this is sort of lost in the activity. And that's shown here in terms of sort of the average response. And another thing to notice is that the response is maintained, sustained maybe for another 100 or so, 150 milliseconds after the stimulus is turned off, okay? So notice, it starts getting, it's on maybe 80 milliseconds after the stimulus is on due to the delay. It's rather high in the hierarchy, and it dies out even somewhat later after the stimulus is turned off. Okay. So just to make the point, and that's relevant, okay, what are the different areas doing? So since my time's up, I won't get into this in depth, but I'll just say that, okay, you can say, okay, well, maybe you could think, okay, maybe one area is sensitive in telling what is the orientation of the face, irrespective of what face is it. Is it head-on or to the profile and so on and so forth. Another is to look at that you'd have specificity for an individual face. So what you could do here is basically look at the activity profile. Okay, so we're going to get into this in depth in the students' talks. I don't want to steal this from them. But the idea is to look at now, rather than the activity of one cell, to look at this across many, many neurons, and to see how similar is the activity profile when you're giving one stimulus compared to the other. So it could be one stimulus in one orientation versus another orientation, okay? Or it could be different faces. And this is sort of something that if we have time after the students finish the talks, I'll get back to this. But it's a similar idea to what you're going to hear. And just to sort of have a brief introduction to the talk, I just want to present this. So the idea is the following. You give two different objects. Say one object is a face, another is this tool here. And if you look across neurons, okay, we don't know how many neurons are participating in this coding. But let's say we've recorded from 582 neurons. Okay, so each neuron has its response to a specific stimulus, right? So let's say neuron number 1 may have responded to this face rather poorly, but also to this object. But neuron number 3 had responded stronger, somewhat stronger to the face than the object, and so on and so forth. Okay, so you get a representation which is based on the vector. That's the idea. We don't know that the brain is using this, but we are using this. We're saying, okay, here we are. We have 582 cells. Here's the vector representation of one image. Here's the representation for another image. How different are they? If they're similar, it's similar objects. If they're different, it's very different objects. Okay? So you can now use this representation. Although for an individual neuron, there may not be much of a difference between them. Okay? If you're now taking many, many neurons, you'll start to see differences because some of the neurons will show selectivity for one object over the other. Now, if you look at an individual neuron, here's the two objects. But as I said, it could be firing to the face and to this plier, and I don't know, to a washing machine. Okay, so it's not a neuron which is sort of a washing machine detector, not at all, or a cup detector. But if it's part of this sort of encoding based on this vector representation, you might be able to tell one object from another. That's the basic idea of this. And now I've finished my time, so we'll hear from the students about this approach. We take let's say, a 10-minute break, and then we have the student presentations. And then if I'll have time, I'll sort of summarize things. If not, we'll just say goodbye at that point. Okay? אז ניקח הפסקה. כן. זה שלך, יאללה. תיקח את האור, תוכל להסתדר, אתה רוצה את ה... אתה רוצה את ה... כן, יש לי. הפוינטר, אתה רוצה גם כזה להזניק את ה... יש. אוקיי, אז זה פוינטר פה. תעביר אותו ל... אוקיי.
לא יוקר. כן, אתה יודע מה?
קונטרול, מה אתה זה לסקרין שלך? קונטרול מה... זה פה? בדרך כלל יש אחד שהוא להעלות את המסך. אתה מכיר? למצגת. פונקשן. כן. יופי. לא, התכוונתי. טוב, הוא כבר מציעה את זה באופן אוטומטי. בסדר. אוקיי. היי, מה הייתה?
זהו, התפשטתי.
מצוין.
לקח לי סופש, אבל אני בן אדם חדש.
לא ישנתי זה לא קצת לילה הזאת. בדיעבד, הפעם הבאה, רק טיסת אין טעם. הטיסה שאני הביא איתה לקחנו יותר כל פעם.
זהו, זה גמר אותי, זה יומיים בלי שנה.
הטיסת היה לשם בסוף. גם בדרך חזרה בעצם לא ניצלתם, נכון? הבנתי שזה לא היה מוצלח למשל. יום שני, כי הילדים כבר מסרו.
יום שני, נכון, יום שני היה לנו רק את עד הצהריים.
כן, סתם, לא
אנחנו נהנינו.
בסדר, גם
הלכנו עם זמן לשוק. זה היה ממש כיף. האמת שזה היה במקום דווקא. זה היה במקום כי עמדנו בלי הפסקה כאילו כל השבוע, ואז זה נחמד כזה שהיה לנו...
100%, לא, רק חשבתי שזה...
מבחינת עבודה זה לא היה כאילו... אבל מבחינת נפשית זה היה...
לא, כי הדרכים ששיטקתם באמצע הלילה.
אני הייתי מוכנה לשלם את המחיר של...
קניתם משהו מוצלח?
האמת שכן. קניתי כזה כמה סובנירים.
תביאי, תראה. תבואי, נו, מחר.
יאללה, מחר. אבל אני כבר אוכלת את זה, לא אוכלת כאילו זה הפרויקט שלנו, המחקר שלנו זה על ילדים באתיופיה שעוברים מחלה ועוברים ניתוח לא יודעת. כן, זהו. אבל זה גם משהו שמאיר רצינות חיים שלנו.
צפתי שקופית סיכום.
מגניב. עכשיו?
עכשיו. אם בא לעבור על זה.
יופי, זה במקום.
איך אתם מוסיפים? אתם מוסיפים שאני לא אגיד מילה כי זה יפריע לכם, או שאתם כאילו, אני מוכן, בדרך כלל אני משתדל לעזור לאנשים, אם זה לא עוזר אני סותם את הפה ואני אהיה בסוף. תגידו אתם מה שתרצו.
אני תודה רבה.
בסדר. אני מקווה שאני כן, זה בדרך כלל לא. כן, לא, בסדר. כן, בסדר. שתי דקות. הגיע המחשב? את לא במשרד. שהגיעו? איזה ביזיון. הכנת ראשון. תשלחי לי את הטלפון? אוקיי, בואו נתחיל את Have a seat, please, and we'll start.
Okay, so my name is Diana, and together with the staff we will present this paper, which title is Let's Read Together. Selectivity and tolerance invariance both increases as visual information propagates from cortical area V4 to IT. So in general, this article will discuss the activity in V4 and will compare it to the activity in IT in two different tasks. So I will start from describing the object recognition problem that our visual system has to face. So when we look at the world, we see different objects. So we need to distinguish between the squirrel and the giraffe, and also the squirrel and the trees, and giraffe and the pole. No, אני לא אוהבת. So this means that our system has to face the selectivity problem. It has to be selective for this configuration and different for this configuration in the world, and also it has to be tolerant or invariant. So the same object can appear in different scales or in different positions or in different contexts. But still we need to know that this is the giraffe, even though all these transformations occur in the world. So one possible solution to this problem is done in, like this is the assumption, that is done through the ventral visual system, the ventral stream, which starts, this slide I saw from Udi. So it starts from the retina, goes to LGN, V1, V2, V4, and the IT. So the assumption is that as the information propagates in the ventral visual system, in the ventral stream, the selectivity and the tolerance are getting more complicated. The selectivity and the tolerance increase as we go further in the ventral stream. So what they did in the paper was to compare the activity in V4 with the activity in IT in the test I will describe later. So when we compare between the receptive fields of V4 and IT, we see that the receptive fields of V4 are much smaller than the receptive fields in IT, and they're encoded contralaterally. Also in IT, but in IT the receptor fields are much bigger. There is much more overlap between the receptor fields, but also there is some inclination for contralaterally encoding. And in the experiment, they wanted to tile all the receptor fields of the cells, so it will match to the area where the stimulus is presented. In both cases, in V4 and IT. So the red circle means the area where the stimulus was presented, and we can see that the receptive field covered both in V4 and IT, the area of the stimulus. Okay, so here we can see the example of the RF of V4 and the area in the brain. So there were two monkeys with 4 chambers for recording, 2 in each hemisphere, one for V4 and one for IT for recording from both areas. And on the left we can see the receptive fields from both hemispheres, how they tile the area of the stimulus where it was presented. So in general, the task was for both monkeys to fixate on the right square, and then once the fixation was done, there was a presentation in succession of different images. Each image was presented for 200 milliseconds, and the monkey had to fixate. But the two monkeys had different tasks, slightly different tasks. One monkey had to identify the image of a motorcycle, and then once he saw the motorcycle, he had to fixate on this spot, which was lower. And then if he managed to do that, he got juice. And the other monkey had to just passively fixate on the images, and once he managed to do this for 2.5 seconds, he got juice. So these two different tasks, basically two monkeys saw the same stimuli, but did a bit different task. They wanted to compare the activity of V4 and IT in response to these images, and to show that it does not depend on the task itself. And this is what they show later. So all the data is from these two monkeys.
So just one thing, there's 2 to 20 images, so they show, just explain, they show this in sequence, and then at some point there's just a motorcycle there, randomly interspersed.
Okay, so the motorcycle was for the second monkey, and the motorcycle appeared randomly in N position between these 20 images. And it was in succession. It didn't stop once the motorcycle appeared. The motorcycle was between the images, and once the monkey saw the motorcycle, he had to fixate, but the images were displaying further.
You had to make a saccade and fixate in a different position.
Yes.
Okay.
This was his response to see that he saw the motorcycle. And also one more detail is that the images of the motorcycle, there was a variance between these images to make sure that the monkey sees the motorcycle and not just a local feature, the wheel. So as I mentioned, the goal of this research was to compare between the activity in V4 with the activity in IT. So they recorded from neurons in populations in both these areas, with a specific analysis, which we started to describe previously. So what they did was to show these different images, and for each image to describe the activity of the population for every image, and to see how different the activity of the populations in different images. So they had a population of N neurons. We can see here, for example, this N neuron, and for a specific image, for example, image A, and there was a specific time window which corresponds to the display of the image, 200 milliseconds, and they recorded from all these neurons and neurons, and they counted the number of spikes of each neuron in this time window for a specific image. So all these neurons together, they create a response vector through the linear classifying Riddle's procedure. And in this vector we can see here. Just to mention that here we see it's two-dimensionally from two neurons, but in real life it's n-dimensionally for comparison of all neurons together. So here we can see comparison of only neuron 1 and neuron 2. So for example, neuron 1 has weak activity, and neuron 2 has a high activity, and this is what we see here. And they have P presentations. So the image A has P presentations, and it created a cloud because there is variance between the activity of the cells. And also M images, so we can see the M images as different clouds in different colors. And eventually they wanted to get a linear hyperplane, which means this line that separates the best between one cluster to all the other clusters. And the analysis eventually was to see if in a specific trial the response of the response vector fell in the correct position, which was determined by the hyperplane, or in a different location, which counted as wrong. Okay, so 80 trials were to train these linear hyperplanes, and 20 trials were to test.
So just a few points. I think one thing to mention is that they don't record the neurons simultaneously. That's very important. They don't record them simultaneously. So this is for neuron 1. So what they do is they move electrodes, just like I explained to you in previous talk. And they record these neurons one by one. So this is actually the firing of neuron 1 for this image. And I don't know, for a different image, you're going to get a different response. So you get the responses for neuron 1 to all the possible images. Then you move on, record from neuron 2. So all this analysis is done offline, of course, after you've gathered your, I don't know, 500 neurons or so to be able to generate the vector. It's better, obviously, if you could record all 500 at the same time. Because this is sort of reconstructed data in the sense that they're from different trials. So just to understand what are the limitations here. That's an important thing. The other point I wanted to make, which I think Dana mentioned very well, but just to illustrate this, is that you use most of the data to train your network. Okay, so you have a network which sort of is trained to say, you take, I don't know, 90% of the trials to say, here's the representation that you get from 90% of the trials to stimulus A, and here's the cloud that Dana said for stimulus B and so forth. And then you test it on the rest, the 10% more of the leftovers. Okay, so this is sort of a cross-validation test. That's used in various fields. It's the same idea. You have the statistical learning that use most of the data to understand what is the sort of plane of responses, and then you leave out some data to test your hypotheses about this. And last thing is it's a linear classifier, meaning that you have these lines here which separate the border from of one object to the other.
Okay, and the equation for the linear hyperplane is, you can see it on the left. Okay, so as I mentioned, the task was to see different images. But also I mentioned that the aim of this research was to test the selectivity in V4 and IT and the tolerance in V4 and IT. Therefore, they made two different sets of stimuli to test these two processing in the areas. So I will start from describing how they tested the selectivity, and the staff will continue with the tolerance. So to compare selectivity in V4 and IT, they used a set of 50 natural images, as you can see here. This counts as a natural image. They extracted from this image 700 parameters, statistical parameters, that describe the local features and the luminance and the spatial frequency. And then, so they had the 700 parameters of the essential features of the natural image. And then they took Gaussian white noise and made many iterations until this noise could fit to the 700 parameters. So eventually they got a scrambled image, which the correlation between the scrambled and the natural was very high in terms of the local features. So this scrambling procedure was aimed to preserve the local features as lines, edges, corners, and junctions, to keep the luminance and the spatial frequency, but destroy the global features. So once we see this image, we can't see something holistic. We can't see the gist, we can't see contours, we can't see shape, only the local features, which is supposed to be aligned to the natural image.
So just one last point about this. If you were to look at, I think Dan mentioned this, but just to reiterate this, if you were to look at V1, these V1 neurons are sensitive to local features. So they respond, the idea is that you want to sort of equate things in V1 so that the image at the left and the right would elicit the same responses from V1 neurons. But then if you move, we are able to say that the one on the left is objects and the one on the right is just texture, right? So something happens at higher levels which allows you this separation. So that was the aim.
Right, and they will show this in the hypothesis graph. So they had 50 natural images and 50 scrambled images, as we can see here. So this was the hypothesis, as Udi mentioned. That in general, what we see here is the performance as a function of the ventral visual pathway. Performance, I just want to remind you, performance of the cells means that percent correct, like you remember, of if the response of the population vector was correct or wrong according to this. So performance is function of the ventral visual pathway, and the measurement was the conjunction sensitivity. Conjunction sensitivity is a form of selectivity that describes the complexity of image features that activate the neuron. So in V1, that is sensitive to simple features as the small lines in different inclinations, we expect to see a low conjunction sensitivity, and in more advanced areas in the ventral visual pathway, we expect to see a higher conjunction sensitivity, which means that to activate the neuron, we need a complex stimuli, and this is what we see here. So early in this pathway, we expect to see a similar response for the natural and scrambled images. So we can see that the cluster of the clouds are separate, and maybe they will change their pattern, but in general we expect to see this pattern separate. And then as we go further in the ventral visual system, for natural images we expect to keep this separation so that the cells know how to separate between these images, but for the scrambled, they don't in higher areas.
Then just go back one slide to make this, okay, this one here. So just to clarify, you're talking about how can you separate these 50 different images, okay? So what Dana was saying, that in IT you're able to separate between these, okay? The question is, first of all, if you compare this and this, they have different local features. So as far as V1 is concerned, it will be able to separate between these as well, right? Because, okay, and that is what she's shown. But as far as IT, maybe you're not going to have sensitivity to these because these are non-natural images. If you're using some sort of a categorization, here's a baby, here's a person, and so on, here's a set of persons here, then clearly you'll be able to discriminate between these, but this is much more difficult. So that's the image that she saw. As you're increasing, you're going from V1, which is here, to IT here, you see that for the scrambled images, you're unable to discriminate between the different scrambled images. But you are able to discriminate between the natural images. Okay, that's the point.
Okay, and then they tested this hypothesis by comparing the activity in V4 and IT. And these are the results they got, which support the hypothesis. So what we can see here is that performance is function of number of neurons. So as the number of neurons is larger, we can see percent correct gets higher. But also that we see that there is a difference between the activity in V4 and the IT. That once the images are scrambled, there is much less percent correct in the IT. And on the right, we can see here is a function of number of neurons. Here we can see just the 130 neurons, the comparison between V4 and IT, and it supports the hypothesis.
Okay, question two. Why when you increase the number of neurons, you are able to do better?
We separate.
Yeah, because the hyperplane, so think, if you start from, let's say, 2 neurons, you have a plane of n of 2. When you're increasing the number of neurons, you have a higher hyperplane, then you can separate these clouds better as you increase the number of neurons.
How did they separate? Just by a linear separation?
Yes, so it's a linear, yeah. Yeah.
Okay, and now, Asaf?
And maybe it is noteworthy that all the manipulations were done on the algorithm and not on the neuron. I mean, as Udi mentioned, they recorded all the neurons at once, and all the manipulation were they did on the algorithm. So
No, they recorded them one at a time.
yes, and also one at a time, but in the same period, and then all the analysis was a post hoc.
Absolutely.
Yeah. I would. Yeah. Okay. So you are already familiar with this slide. So each object may have like a few versions in different conditions that we want to have the ability to generalize the object over the across the conditions. So as Diana showed, it contains like over scales, different positions, and different contexts. So what the researchers did now is to check whether if you show IT and V4 compared to V4, you show them a reference object which is in black. So how well are they, the neuronal population, in generalize the object to also the other conditions. So similar to the hypothesis that Diana showed before about the selectivity of IT compared to V4. So we have a kind of opposite hypothesis about the generalization. I mean, V4 was much better in sensitivity to the scrambled object, and IT is supposed to be better in terms of generalization. So this is also like an ideal hypothesis of advancing in ventral pathway. So okay, yeah. So as you can see, the black dots dots are the reference, and the blue dots are their modified version.
What do you mean by reference? Say one of the pictures. Let's say an object.
Yeah, it always was this
The giraffe, for
object.
instance. Okay. So now you're asking if you change the giraffe, moving closer or further away from it, then the giraffe scales. Right. It's not that you have a toy giraffe, but as you're moving closer in terms of the retinal image, it becomes bigger. Right. So we want something which is sort of a giraffe representation, which is scale invariant. And so if you sort of
Yeah.
go.
So exactly. So V1 does not know nothing about giraffes and objects. So he's supposed to do really bad at this task, and will have like a coding representation similar to this plot, and then he will not have the ability to linear separate between the all objects, because if he will separate the reference, so its modified version will be also in the other side of the line, but IT, or more advanced and elaborated population in the ventral system pathway will have the ability to generalize and actually to put both the reference and the condition and its modified version on the same side of the line. Okay, so they tested this hypothesis and did the analysis, and they got it. They got a successful result. So as you can see in IT, in V4, the difference between the conditions and the reference was quite high, and in IT, so it did much better performance. You can also see that in those two graphs that again the performance got better as a function of the number of neurons, but in IT it continued to increase also to 140 neurons, which was the maximum of recorded neurons, while in V4 the increase is getting much less. It cannot pass the threshold of it doesn't get any better. And here you can see by detail for each condition how good the generalization was. So actually the researchers do not talk so much about it, but I mean they just did the overall statistical test and showed that it is significant, as it can be really shown here. But they didn't talk about the differences between conditions, which can be quite interesting. Why, like in scale, they had in little scale, small scale differences, they had no difference at all in between generalizations, and here they had very big difference in position, but little position less difference. So
let's think about it for a minute, since you've raised this issue. I'll raise this as a question to everyone. I mean, I think that you could easily understand why you're going to get a greater position invariance in IT than V4. Why is that? The simple reason that we've just shown in the introduction. I think that receptor fields become bigger, right? So receptor field becomes bigger, it's position invariant. So no surprise if you're looking at V1, the receptor field, once you are out of the receptor field, you don't get a firing from the cell. So clearly, the position invariance is sort of an immediate consequence of the fact that receptor fields are bigger. What is more interesting is the context. Okay? So context is not trivial, right? That you're going to, it doesn't matter what the context is, you find that in IT you get much more generalization than in V4. Okay? And in terms of scale, that's another issue. Does it matter if it's closer or further away from you? Okay? Again, when it's closer to you, then the stimulus becomes bigger. So it gets out of the receptor field, right? So that may not be such a surprise. The context, I think, is the most interesting, and and somewhat surprising result. And it's not nice to think about this again. I mean, when you're moving from a vector representation to think about this again in terms of the receptor field and single unit properties to understand some of the features.
I think they tried to overcome the triviality of
Not true. I mean,
no, the triviality of the position, and they adjust the degree to the receptor field size. So somehow.
Well, no, receptor field size changes also within an area. If you go back to Diana's presentation, you show that it's quite a bit of a change. And I think what they do, it says here plus minus 1.5 degrees, so that's what they do. They move it by 1.5 degrees or 3 degrees, okay? And so, I guess you're right that it's somewhat trivial, but it's nice to show this. I mean, okay?
Okay.
So some aspects are something that you can immediately think about, and some are less sort of obvious. And I think that is the distinction I want to make. Go ahead.
Okay. Yeah, so I will go over it really briefly. So then they asked, okay, maybe V4 does not have the ability to generalize without constraints, but it will have the ability to generalize with constraints. Namely, when you train the algorithm not just on one reference, but on all the conditions together, so then maybe you can get the linear separation you accepted, and in these terms, so V4 will be as good as IT, or maybe almost as good as IT. So they ran this test, and they didn't find so much difference between the tests. Here you can also see the supremacy of IT comparing to V4. So it's not a matter of constraints, just V4 really does not have the ability to generalize in any sense. I mean, not like IT at least. Okay. And then they asked, okay, but we measured still now just the populations, but how the results of the population, how does it come, how single neurons can be responsible for the differences in the population. So to measure this, they did an analysis on single neurons, and they did it with a little bit different way. What they did was to compare between the true positive of each neuron and the false positive. We have seen something very similar.
It's called hit. Y-axis is hits, and the x-axis is the false alarm, right?
So hits and false alarm. So if the neuron is completely random, so we're supposed to get this, like the result on this diagonal, and then after integration, so the area here will be half. And if the neuron is ideal, so we're supposed to get like this step function along the axis. So the integration will be 1. So and then they computed the value for this value for each neuron. That's what you can see here on the x-axis, and then they compare it. They try to correlate it with the behavior of the neuron, of the subpopulation of the neuron. So they did a quite complicated procedure about that divides the population, the whole IT and V4 to subpopulations, and then ask how the performance of each subpopulation is correlated to the performance of a single neuron. And what you can see is, okay, first of all, that it is high correlated, both in V4 and V4 and in IT. And what more can be seen is that IT also in this condition has a much better generalization ability, because you can see that in the scrambled objects do not pass like this border. That they are not, they cannot do better than 0.65 or something like that in in the rough analysis, while the natural object does have the ability to get much better performance, and before you cannot see exactly this. You can see a tendency, but not in the same values.
Okay, we'll cut here just so we have time for the second group to be fair for them. But can you just summarize? Okay, so what do we see here? Basically, okay, we've gone for the ROC analysis. I think you know this, but the ROC analysis is done for what? What are the images that they're using? That's what I'm interested in. Okay, so you're saying here, you've shown here one example of I don't know, one neuron. Doesn't really matter what sort of algorithm you're using there, but okay, you get this sort of neuron which has. So remember, okay, what are the numbers here?
The area on the
So it's between 0.5, meaning there's no information at the level of neurons, and 1 is maximum information, right? And okay, so what do you do here? You sort of maybe help me here, but you're sort of looking at.
It's supposed to be 0.5, 0.6.
Yeah,
it's just split.
Mistake here.
It's split.
Yeah. So some neurons are more sensitive than others to the different images, right? So they do the ROC sort of comparison between two different images.
I forgot to mention that they did not one against all comparison. That's what they did like at the other analysis. But here they did pairs of objects and just compared between each object to its pairs.
Okay, let's get to the gist of it. The question is, some neurons are more sensitive to the others, right? They're more sensitive to tell. Okay, and the question is, okay, if they're more sensitive, what do you expect? That they're going to have a bigger weight in the decision about is it object A or object B, okay? And I think that's the analysis was to look at that in terms of the single neurons. We don't have much time to get into this, and so it's a side point to the main point that we wanted to just want to summarize here. That okay, what we've seen here was that if you look at the population of neurons. You have a summary here, which I think you wanted to make. Okay, so go to the summary.
Okay, so
skip this and let's just go to the summary. And you explain. You give the summary. Okay?
Yeah. So as we saw, so IT has a better generalization ability than before. We also saw that that a single neuron performance is correlated to the performance of the population. Although they do explain that it cannot be responsible for all of the difference of the population. I mean, it is not just the sum of the single neurons. It has like the all is greater than its
yes.
Shalem Gadol and this improvement in performance is also correlated to a few parameters which we didn't really saw, but very interesting to the receptive field size. We talked about it a little bit, and the number of spikes and etc.
So the number of spikes has to do with sort of the I would say the range at which the neuron fires. So if its maximal spike rate is higher, then of course you have a higher dynamic range to play with, and of course then it's no surprise that the neuron is more sensitive. Doesn't have to be this way, but it's generally the case that the higher the firing rate for the optimal stimulus, the more you get a discrimination between the different spin bands. Okay, so that's thank you very much. Very nice. Okay, the questions, if we have time, we'll leave for after. I just want to give a chance for the other to give the presentation. So go ahead. So the next presentation we're going to hear. So as you set things up, I'll just give a little an introduction. Basically, what we see now was computation based on the population response of neurons. Okay. Now, the next thing we're going to see is work that has been done by Nicola Kritzkortev, which is, I find, it's 10 years ago, but it's extremely interesting because what he's done is take, on the one hand, just like we've seen here, a different population of recordings done by Keiji Tanaka's group, where they've sort of shown different images of objects and look at the population response of, I don't know, 500, 600 cells in a similar way to what you've just seen. On the other hand, Jacob Krikskot himself with Peter Benedetti, what they did was use the same images with humans. Now, they're not recording from single neurons now, of course. What they're recording is from single voxels. And each voxel, okay, is of the size of, I don't know, at the time that was about 3 by 3 millimeters. In 1 square millimeter of cortex, we have something between 50,000 to 100,000 neurons. So if we're talking about 3 by 3 by 3, that's 27 millimeters square. So we're talking about something of the order of, between 1 and 3 million neurons. Okay, so looking at the average signal of 1 to 3 million neurons. Another point to mention is that this is not just in space that you're averaging, but also in time that you're averaging. Because remember that the activity of the fMRI activity, the bold activity, is lagging, and it's on a much slower time course than the neural activity. And so there we're trying to do the matching between them, and we'll look at the results, and then we'll have hopefully a short discussion about it.
So, hi, I'm Shachar, I'm Physiology A, for those who don't know us. And we're going to present the article, Matching Categorical Object Representations in Inferior Temporal Cortex of Man and Monkey. It was published at Neuron in 2008 by people who I don't know how to pronounce, so I won't. So, as we learned from Udi's lectures, after the visual input arrives at the cortex in V1, it continues propagating across multiple cortical areas in 2 main pathways. The first one is the dorsal stream, which, as we saw, is dealing with visually guided action, motion perception, and localizing objects in space. And the second pathway is the ventral stream, which is more connected with object recognition and perception of shape. This pathway carries information from the early visual cortex through V2, V4, and finally arrives at the inferior temporal gyrus, or as we call it, EIT, which also contains the FFA, the face recognition area, and the parahippocampal place area, which involves environmental scene recognition. Okay. So as the title suggests, we are comparing human and man and monkey. So why even comparing between them? In previous studies, there were evidence of coarse homologies of the early visual areas. In both species, the IT is object sensitive, and this type of interspecies study can contribute to the way we make deductions about humans from animal studies. It can also propose an efficient way of comparing between species, and overall has implications about visual perception and systems neuroscience in general. So the challenges that arise from this type of comparison are, since we compare different species with different brain properties, the comparison cannot be direct. The difference of species requires us to use different measuring methods, and that poses another challenge. And since the measurement methods are different, it creates a scaling problem, which we will mention later. So the research questions that this study has dealt with were, do monkey and human IT response patterns to stimuli from form categorization? How similar are the response patterns of human and monkey IT between the species? Does IT keep within category information of exemplars? Namely, does the IT care about which exemplar is presented if it is from the same category? How IT object representation is transformed from the early visual cortex? And finally, what low-level features computational models can account for the IT categorization? So the experimental design. In order to address those questions, the researchers designed the following experiments. In both methods, the task was a fixation task. 92 isolated real-world objects were flashing rapidly in a random manner. For the monkeys, it was a passive fixation, and they received juice incentive. They had two animals, 674 neurons were coded via single-cell recordings from both animals, like the information of the whole neurons was from both. The stimulus duration was 105 milliseconds, and the interstimuli interval, the SI is not inter-spiking interval, note that, was 0 milliseconds, meaning it just switched the object. And for the humans, they had 4 participants that performed some sort of discrimination task, while in the fMRI, 316 voxels were gathered from the IT, and the duration of the stimulus was 300 milliseconds, and the interval between stimuli was 3.7 seconds. So these are the stimuli. You can see the animate stimuli on the left and the inanimate on the right. Within animate, you can see different categories, and also in the inanimate, there are categories of separation, although this separation to categories was not present in the actual task, and it is just a way to sort the stimuli. Now, Elio.
Okay, so they measured the response of neurons in the monkey and the box of the human, but to different stimuli in the IT. What they wanted to do is to be able to compare between a representation of different stimuli. Okay, to be able to compare the response pattern of stimulus A to the response pattern of stimulus B. And what does it mean? A response pattern is just as Diana said, it's the same here. The response pattern is a vector, and we have for each stimulus we have an array of responses of different neurons, and for each neuron we count spikes and sum them and average across repetition. So for stimulus A, this is the vector of response. Also for stimulus B, stimulus C, etc. So we have a matrix of the whole stimuli with 92 stimuli and the number of neurons. And we get this matrix that each column is a response pattern to a stimulus. Now in human it's the same, just instead of neurons they had voxels, fMRI voxels with the mean activity of the voxel. And next, what they wanted to do is to compare this to this, and the response pattern of A to B, A to C, B to C, etc. So we have 92 or 92 correlations between the response patterns, a symmetric matrix of 92 or 92, and that's what the researchers did for the monkey and for the human, 2 different matrices. Just instead of
Wait, just one thing. What they don't do, can you go back? Look, but there's some variation here. I mean, you look at this neuron, you get, okay, it fires, then there's a period it doesn't fire, then it fires again, so on and so forth. So that temporal variation is not taken into account. What you care about is what is the average firing rate during the presentation of the stimulus. And so that's relatively low here. I don't know, here maybe it's higher, so this is more white. And this is still higher, so this is very white. Okay, so you don't take into account the temporal variation, which may be very noisy also. Sometimes, but there could be more information in sort of the temporal development of the firing, which this model does not take into account.
right, they take a window and they don't care about the temporal resolution. And okay, so they get a matrix. Just instead of computing Pearson's R, this presents the 1 minus R in percentile. So this matrix for each species. And what do we see here? It's a 92 or 92 symmetric matrix, and that each pixel here represents the correlation, the similarity or the dissimilarity between two objects. So between the response patterns of two objects. So for example, if I look at a pixel here, I can see the comparison, the level of similarity between these two objects, between these two spaces, and it's blue. It's very similar. But if I look at another pixel, I can see that response patterns for this space and this road sign are very dissimilar. Okay? And as we said, this is a symmetric metric. We don't have it to have a 92 comparison. The information in the upper triangle is the same as the information in the lower triangle, and the main diagonal is all blue. We understand why.
It's correlation of itself.
Right. Line of zero, because these are the correlations between a stimulus and itself.
Is it brain activity or just an algorithm?
This is just it's normalized. It's in percentile. This is very dissimilar relative to the other comparisons here.
No, they ask you to based on brain activity or just an algorithm, okay? Of course it's based on
brain activity in
No, but let's understand, okay?
Yeah.
You say brain activity. What do we mean? You take the vector for a given stimulus, okay? A, let's say, is the one phase, and B is another phase, okay? And you ask, what is the similarity between these two vectors, okay? They maybe have n neurons here. You look at this. Well, these two are very similar to one another, right? So they have a high correlation between them. Well, and A and C are somewhat more different, okay? They have a lower correlation. If you go back, you go to the next one, just to clarify. So that's what you saw here. And just to make the point that there's some loose ends here in the sense that, okay, if you notice here, it's not R that they show, it's 1 minus R, okay? So it's the dissimilarity. R would be the similarity, 1 minus R is the dissimilarity, but there's another trick here, because they divide it into percentiles. I think that's what you heard just now. So 0 would be the most similar. So you have just for each entry here an R. So I don't know, you get for this one an R of 0.25, and you get for this one an R of 0.63 or whatever it is. So you rank order them from the highest correlation to the lowest correlation. So 100 would be the lowest correlation between them, and at 0, so the percentile, 0 would be the one with the highest correlation, okay? So we don't know what the value of R's are here, but they're already sort of presented as percentiles. They maintain the rank between them, but we don't know what the values are in this. So there's a bit of a trick here, and that's also used to compare this.
Okay, so as I said, they created one for the monkey, average across the whole neuron, and average across the two monkeys, and one for the human. And these are the actual results, and this is the human matrix, and this is the monkey. And we can immediately see some obvious things that pop out. And can someone tell me the main thing we see here, or Udi wanted to?
Yeah, just to mention again that the human IT is not based on neurons, right? It's based on these whatever
Right.
300 voxels or so that they have from human IT. Okay, and so you can notice how similar they are, right? I mean, that's quite amazing that you basically get the same pattern, right?
Exactly. As Udi said, the first thing we see is that this is very similar, and it's not obvious for two reasons. One, that this is from neurons and this is from voxel. And one voxel can reflect the activity of hundreds of thousands of neurons. And the second thing is that this is monkey and this is human, and we talk about object recognition, and we could have expect to have different representation for objects in different species. So this is non-trivial, and the second thing we see here are these boundaries. We see a clear distinction between animate objects and inanimate. We get very high correlation, I mean relatively high correlation for animate versus animate, or for inanimate versus inanimate, but very low similarity for comparison between animate and inanimate objects. And the third thing we see here are these squares that reflect the similarity between patterns for faces. And not only for between human faces, but only between human and non-human faces. Okay, so this is what we said. Very similar matrices, animate and animate distinction, and very high similarity between faces. But yeah.
Non-human are just monkeys?
Faces of monkeys, lions, animals.
Also, because it's quite strange that also the monkeys have a better correlation in the human faces than the non-human faces.
It's not very surprising actually, because we can distinguish between faces much between human faces in a better way than maybe monkeys do.
No, but you can say that a human too, that monkeys cannot discriminate very well between humans.
Yeah, all humans
Okay.
look the same for them. Maybe.
Yeah, but it's not the result, because they have
Why? Better correlation between
Human.
faces means that I don't distinct between the faces, maybe. And this is the monkey. The monkey, he has a blue square, okay? But
No, so put it in another way, it's surprising that you don't see this in the human data, that because we are able to distinguish between the various faces in humans. How come are they all similar to us, okay?
yeah, right.
And so the answer is, maybe, well, what is the answer? That maybe it's not a sensitive enough measure for this, okay?
Right, we talk about the whole IT actually, and
We talk about
the fusiform area is inside, but I mean overall the activity pattern for of the fusiform area can contribute to the overall activity of the IT. So maybe the fusiform area distinct between different spaces, but it always contributes to the overall activity of the IT when we see spaces. So we see similar, I don't know. But just by looking at this, we can't say for sure that categoricality is the main factor that determines the response pattern in the IT. I mean, we see it here, we see the boundaries, but this is a 92 or 92 matrix that we have, we can arrange in different ways. And when we arrange it by category, we see this boundary, but the categoricality itself didn't emerge from the data. So in order to test it, what they had to do is to do a multidimensional scaling. And what this means, what multidimensional scaling, the main idea behind it is to take the multidimensional vectors we have. I mean, every stimulus we have coordinates relative to a lot of neurons, and to take this and project it on a two-dimensional space in a way that the distance here between stimuli reflects the similarity between them. That's what they did here, and we can see the arrangement for the human and the monkey, and we see that the main grouping here is according to the animate-inanimate distinction, and also inside the animate there is a grouping of body and face, both in human and monkey. So here we can know that the categoricality arises from the data.
So you guys have heard about multidimensional scaling? Is that clear what they've been doing here? Let me refer, we don't have to cover everything, but let's understand this because this is important, right? So you think about this. Each image here generates an activity pattern across these n neurons. There may be, I don't know, 600 neurons or so. So you have a 600 dimensional vector. describing the firing or this image generates activity, I don't know, 20 spikes in neuron number 1, 13 in number 2, and so on, and a vector of 600 such neurons. Another image generates another vector. So we're talking about a 600 dimensional space in terms of, in terms of the firing elicited by each of these symbols here, or these images. Now, of course, you cannot describe this, you cannot describe these 600 dimensions, so we want to reduce this to a description of 2, maximum 3-dimensional space where we'll be able to see these clouds, and then now we can compare the distances between them. So closer clouds have higher correlation, similarity, further away clouds have lower similarity. And that's what's being done here. So there's a way to do this called multidimensional scaling, basically reducing the dimensionality with minimal warping of the data. So and once you do this, this is what you get. And the nice thing is to look at this and compare this with this, and you now have the ground.
Okay. And these are still the same arrangement, the human and the monkey. And here each pair of linked dots represents the same stimulus in monkey and human. For example, this is stimulus A, this is also stimulus A, and the fiber that connects them represents the level of similarity between the representation between species. A thick fiber means very high dissimilarity. Right? The thin ones are the ones that represented similarly across species. And how do they compute this similarity? It's just for this stimulus A here has a vector of correlations with the other stimuli in monkey, in human, and it also has this vector in monkey. And the extent to which these vectors correlate gives us the similarity of representation across species. And we can see a lot of thin fibers here that give us the intuition that maybe not only category is what determines the representation in the IP, but maybe also there is continuous representation, information within categories that is also consistent across species. This is another way to show the data. I don't want to get into this too much, but we can see that the top level of the hierarchy of this is the correlation. It starts from the low level and go up, and the top level is the distinction of animate and inanimate, and there's this distinction of body and face, and there are also other levels of correlation between stimuli.
So this is like an evolutionary tree, going from the furthest distinction between the two, which is at the top, to the lowest distinction, right?
Yes.
And basically, the point to see here is that they color them according to the categories. So you see that there's generally these are 3 different ways of depicting the idea that basically the categories are mapped very similarly in monkey and in human. So very similar in monkey and human, although it's a different scale and a different species.
Right.
So we have time now for like 3 minutes to conclude everything. I know you didn't have time to get into everything, but that's fine. I just want to understand. So you can have one more slide, and then we go to a
maybe
conclusion.
we'll go to the summary.
This slide wants us to say that
there is a continuous representation within the IT, which sort of the IT cares about how similar the objects are. Like if they're face and 2 faces, or face and sheep, then the 2 faces are much more correlated.
So each dot here corresponds to the similarity between 2 images, and the y-axis is the human, and the x-axis is the monkey, So if they were the same, they should be along the main diagonal, right? And then you see that they're generally, you see some matching between the 2, and then the colors correspond to the specific category, So And you can look at within category, that would be the red cloud, and between categories is between the colors, okay? Just to get an idea.
These are subcategories, but we don't have time.
Yeah, we don't have time.
And like you see that the early visual cortex is very, like, it doesn't look anything like the RDNs we saw earlier, so it's the categorization does not emerge from the early visual cortex, but maybe the continuous categorization does.
Okay, so cool. This is, remember we talked about in the first lectures, we heard about, you know, the differences between early visual cortex and higher visual cortex in the sense that you get sort of sensitivity to categories. And you do this in humans, right? You get the same, now you see no structure at all, while in human IT you saw a very clear structure. So that's nice that you have a similar correspondence.
And they did the cluster analysis for the early visual cortex as well. And they actually took all of the analysis that they did on the early visual regions and the IT in human, and they tried to find some modeling that can account for that from the low level features, and they saw that no models that they checked could account for that. So just to summarize, the IT response patterns for objects cluster according to the same categorical structure in monkey and human. Within category, IT represents exemplar information, which is also consistent across species. Categoricality appears to arise in IT and not in early visual cortex. And low or intermediate level computational models cannot create clusters observed in the IT. And now we left some very small things for
No,
discussion.
we have time, but we wanted to discuss the
Do we have time?
2 minutes, 1 minute, yeah, okay.
So let's only talk about the scale difference. The data of the monkey was based that the minimal unit was the single cell, while in human it was the voxels, and these are very different in terms of scaling. And we got similar results, and so that's surprising. But what does it mean, and how can it be? So this article doesn't address this question, and they don't presume to say anything about the spatial mapping or spatial organization inside the IT. But we thought that maybe we can think about it as we learned in these lectures about the effect we saw in MT about the correlated versus uncorrelated population. And since here in the IT we have columns we have columns, and similar closed neurons have a similar activity. So maybe this what gives us the similarity between imaging a box cell and recording a single cell. Maybe that's something that can be.
Yeah, okay, so that's an extremely important point, because, I mean, first of all, the scale of the voxel is much larger than a single column, okay? So within a voxel you have quite a few columns, okay, around tenths of columns, okay? Because a column, its width is about a few hundreds of microns, and the voxel is about, you know, a few millimeters, 2 millimeters by 2 millimeters. So that's, I mean, you're right that it goes along this, but it cannot explain it completely. I think there's another, I'll take over here because we really have to finish Latin. And I want to leave you with another point beyond the scale difference. I think that there's, the amazing thing is a species difference, right? Because when you see these images, maybe you could go back to the first images. Exactly. Go back to the first images where you showed the very first slide where you showed the different, this one, okay? So you think about this, okay, what happens when a monkey sees this? Does he know that that's a pistol? Of course not. Or this is a plier or something? So, okay, this division between natural and artificial objects makes sense. But within objects, okay, I mean, we tend to, we cluster all the signposts together, and at some level you can say, okay, there could be objects that are visually very different from one another, but they have a similar function, right? So we tend to cluster them together. Well, how does that come about in the monkey? So there's many questions that come up here in terms of how, Another thing to notice is you can classify objects as being similar or dissimilar depending on what the task is. Or so in some cases, if you have to judge their function, it's one thing. If you have to judge the visual attributes, it's another thing. Okay? So this raises, I think this article is extremely interesting, raises a lot of questions which people have started to try to deal with in a more recent past. I didn't have time to get into this, but I think this is a very interesting venue to take. And the other point to mention is, okay, I think I mentioned this, and I have a keen interest in this. It also shows that functional MRI is a reasonable, not only a reasonable, but a scientifically viable technique to use, right? First of all, we use functional MRI to locate voxels which are phase selective and record it from them. And second, you can basically replicate the findings that you find with single units with functional MRI. So it raises big questions, but it's also pointing to the fact that, I think you hear in elsewhere sometimes at functional MRI, you don't record the neurons directly or something like that. That shows that, okay, you have to give it some respect. Okay, I think I went over time. Thank you very much for the speakers. Thank you all. And I think the next, I say goodbye here, and I think I'm passing the lectures to, I don't know, Idan? You've heard Idan. Okay. Whoever it is, it's not going to be me, so thank you. יופי.
אולי אני אסיים את זה. יאללה, תודה. ביי. אז יש לך, נגיד, תאים 1, 2 עד אין תאים. ואז יש לך איזשהו וכל פעם שאת מציגה את האימג' הזה, את רושמת מהתאים. פה יש פעילות כזאת, פה יש פעילות כזאת, ופה יש פעילות כזאת. וקטור, זה אומר בעצם, זה אומר שיש לך, נגיד, איזושהי, בדיוק, כן, יש לך איזושהי שורה של ערכים, וזה כולל כל הדברים. אז זה נגיד נכנס לפה, זה הערך הראשון בוקטור, זה הערך השני, וככה, ואז בסופו של דבר את יכולה ליצור מזה איזושהי משוואה מתוך הערכים האלה. אז בעצם וקטור חי מתאר פעילות תאים לתמונה אחת משותפת. אז נגיד איך יראה זהו, אז מה שאצלי, מה שהראו זה שלקחו בתמונה שאני הראיתי, פה אני אומרת זה וקטור של n תאים, אבל בתמונה שאני הראיתי זה היה וקטור של 2 תאים. ואז זה נגיד תא 1 וזה תא 2, ואז הסתכלו על תא 1, ספרו כמה ספייקים. תא 1 עבור אימג' A היו מעט ספייקים, ובתא 2 עבור אימג' A היו הרבה ספייקים. אז בעצם לוקחים את 1 ו-2, מכניסים את זה לתוך וקטור, ומקבלים את זה, זה וקטור 1. ואיזשהו ערך שמקבלים מזה.
זה תחום שבוע.
כן, אבל בעצם זה נקודה. כן, בניסוי שלי זה נקודה, הם מוצאים איזשהו ערך מתוך זה, אבל פה זה 2 תאים. אבל תחשבי שבגלל שיש אין תאים, אז יש אין מימדים. פה יש רק דו-מימדי 2 תאים, אבל באין מימדים
יש לו מימד Z, כאילו.
לא רק זה, זה כאילו יש אין מימדים, כאילו המון מימדים, שבכל מימד זה משווה פעילות אחת לפעילות אחרת, בעצם ההשוואה של כל הפעילות ביחד, זה נותן ערך מסוים. ואז מקבלים המון ערכים עבור אימג' מסוים, וזה הענן, ואז מקבלים עננים כאלה, ואז מחשבים איך הענן הזה מופרד משאר עננים, וככל שהוא יותר מופרד זה אומר שהתאים יודעים להפריד יותר טוב בין האימג' הזה לאימג'ים האלה.
וזה נקרא וקטור.
פה יש המון וקטורים.
המון וקטורים בתוך הענן.
בדיוק. כל נקודה כזאת זאת חזרה, זאת תצוגה אחת
של אחד זוג תאים.
פה זה זוג תאים, אבל כאילו בשביל לעשות אנליזה של כל התאים ביחד עבור תצוגה של אימג' אחד. וזה כאילו אימג' A, ונגיד אימג' שיוצג פעם 1, 2, 3, 4, כאילו זה כל החזרות על A.
זה N.
לא, זה אימג' A, זה אימג' B, C ו-D, וזה חזרות על אותו.
חזרה של אותו A שהיה אותו אימג'.
כן.
אז פה יש אמצעים, נקודות.
כל נקודה כזאת מייצגת פעילות של N תאים. N תאים נמצאים בתוך הנקודה, והנקודות עצמן זה החזרות על אימג' A.
חזרות על הבנתי, אמצעים זה כל נקודה, אתה אומרת?
כן, כל נקודה כוללת...
למה יש הרבה תאים? אז למה יש הרבה נקודות?
כי יש המון חזרות על אימג' אחד.
זה כל אחד.
Par un analyse, je pense qu'on s'en sort. En considérant que d'abord, le technologie, c'est que si tu fais l'intégrale entre 0 et plus massif, tu as ton Z dans ce format. Il faut d'abord dire que t'attends d'abord
כאילו זה מה שמופיע אצלי, ואני מניחה שזה העיקרון.
העיקרון, כן.
בואי, מי ילך לך? אני לא, זה לא ממש תשובה עכשיו.
Et sinon, j'ai des trucs comme ça. Soit tu modifies après. Après, t'as vraiment donné le truc, j'ai un peu réécrit. Il y a même à un moment donné, je me demandais mais j'ai pensé que tu l'écris que. Oui, oui, moi aussi je le sais, mais je ne peux pas les écrire. Bah, il est chez où, mais en même temps, je me dis, dans un cours comme ça, ça fait un billet de...
באופן כללי, גם וקטורים, לי זה התחדש ברגע שהתחלתי ללמוד תכנות, כי עד אז זה לא התחדש לי, אבל יש וקטור ויש מטריצה, וזה כאילו עוזר לחשוב על זה בצורה ויזואלית, שווקטור זה קיבוץ של מספר ערכים שזה שורה אחת, ומטריצה זה אחד מול השני. אז כאילו פה זה וקטור, כשזה תמונה אחת בווקטור, וכשאת משווה תמונות שונות, את יוצרת מטריצה, שזה שילוב של המון וקטורים ביחד.
Yeah, I think we're going to try